All right, we are going to continue the lesson that we were in the midst of doing yesterday. Yesterday we talked about just DNA, and so today we're adding the idea of something called RNA. And you might say, oh, well, it has sort of the same abbreviation, we just changed the first letter. And so in the name RNA, the N and the A still stand for nucleic acid. Just like in DNA, it's deoxyribonucleic acid. The R here is almost the same. We're just taking off the D part from DNA. DNA was deoxyribonucleic acid. RNA is ribonucleic acid. It's almost the same word. It's actually shorter than the word for DNA. This is another chemical molecule, another little nucleotide that was isolated by Levine. So the same guy from yesterday, Phoebus was his first name, which I find hilarious. He's isolating these chemical structures. RNA is built basically the same way DNA is. It's got a lot of the same basic parts in its structure, but with three key differences. So other than these differences, everything else about RNA is just like DNA. The first difference is that the sugar in RNA is ribose rather than deoxyribose. That's why the name is different. So if you recall yesterday, and I'll just back up for a second, these were our nucleotides, and the sugar was this little pentagon with a D in it. We're still going to have a 5-carbon sugar, but it's a different type of sugar. It's ribose instead of deoxyribose. You might even remember in the video yesterday, as the girl was explaining, she said it's missing a hydroxyl group in DNA, and in RNA it's not. It's almost the same sugar, except for one little piece that's attached. So RNA has a different sugar. It still has a sugar, but it's just a slightly different sugar. The second thing that's different about RNA is that RNA does not contain thymine. There's no T in RNA. But rather, it has a slightly different nucleotide called uracil. It looks just like the other nucleotides, phosphate, sugar, nitrogen base, but the nitrogen base is uracil instead. Okay, so same basic structure, but a different nitrogen base. If you look at my little cartoony drawing of RNA here, you'll notice that there's a backbone. And if you look at the picture from yesterday, which I'll just back up to, right? Same backbone, same alternating structure, that's representing the sugar and the phosphate. So RNA still has sugar and phosphate as a backbone. Now look at the shapes, especially in this top picture. There was like a pointy one, and then its exact complement. And then there was a rounded one, and its exact complement. If I look at the structure of RNA, see how I have the same basic shapes, except the pointy one has a little piece cut out of it. That's to represent that uracil is very, very similar to thymine, but not exactly the same. It's similar enough, though, that the little pointy tip here would still match up perfectly with that other nucleotide that's its complementary shape. So anytime we have RNA instead of DNA, T doesn't go with A, instead it goes with U. Uracil and adenine are close enough in shape that they can both be complementary to thymine. Okay? Then the third difference is that RNA has a single strand. That's a big deal. This isn't just an incorrect picture where I've only drawn half of the RNA. RNA only has one strand. There's no second side. And you might be asking yourself, well, that's weird. How would we make complementary things then? Well, we're going to get into another part of the story where you'll see why RNA needs to only be one strand. But these are the three key differences. Other than those three differences, everything is the same. They both have phosphates. They both have sugars. They're both made of nitrogen bases. They both have A and C and G. It's just the T and the U that are different. DNA is double-stranded, has 2 sides. RNA has 1 strand. Now, does anyone want to ask anything here? Now, visually, the 1-strand thing is the most obvious. But if I had to pick out of these ones, which one is probably going to help you on a test the most, it is number 2. The fact that RNA has uracil, a U, not thymine, the T. There will be a lot of questions on tests where some of the choices will have T's and some will have U's. And if you know which one goes with RNA and which one goes with DNA, you've already knocked out half of the incorrect choices. Anyone want to ask anything there? Now you might also be saying, hey, this is Baxter. It says RNA, then it says mRNA. What in the world is that little M for? There's actually more than one type of RNA. We're not even going to discuss all of the types that exist, but I would like to tell you about a few of them. So the M in mRNA stands for messenger. mRNA is messenger RNA. Like its name suggests, it carries messages around. It looks like a ribbon. So this would be another representation of messenger RNA. The strand on the bottom, that's like the backbone where the sugar and phosphate is. And the different colored pieces that are sticking out, those would be the nitrogen bases. Now, it is a product of something we call DNA transcription, and we're going to talk about the details of that in our next class. It carries the code for making proteins by translating the genetic code. And so one of the things that will be kind of the focus of the next few days is how DNA carries the genetic code and how your body actually gets that information and does something with it. So messenger RNA, it's going to go get information from the DNA in your nucleus and take it to another part of the cell so that stuff can get done with the genetic code. It's the messenger. There's also something called ribosomal RNA. Perhaps you remember that there's a structure called a ribosome in a cell. Maybe you're like, I thought ribosomes were those little dots. They were like little dots on the rough endoplasmic reticulum or little dots in the cell. And that's how we draw them if we're looking at them from really far away. But if you zoom in, ribosomes are actually 2 chunks that aren't super circular in nature, but from far away they do just look like a little dot. And each of them is mainly composed of bits of RNA. Lastly, we have something called transfer RNA, or tRNA. They kind of look like a clover leaf, not a 4-leaf clover, but if you look at the top and you kind of draw around it, it sort of looks like a 3-leaf clover. What it really is a piece of RNA that's been bent and twisted around. And then at the bottom of it, there will be some pretty specific nitrogen bases sticking out the side that will help determine which amino acid we're going to use to make a protein. You'll see this, but foreshadowing, kind of the whole point of your DNA is to tell your cells which proteins to make. That's how stuff gets done. And so the messenger RNA takes the information from your nucleus, brings it to a ribosome, and then in the ribosome, the transfer RNA transfers amino acids, which we put together to make proteins. That is the big picture, okay? Now, we're going to talk about the details of that process. Like, this is just like the intro preview to this, so you know what the types are. Does anyone want to ask anything about RNA? If you kind of glanced behind my scribbles, you would see that the 3rd question on the daily review was about RNA as well. And it asked you to compare the structure of DNA and RNA. That is a typical question. Like, hey, here's some characteristics. which ones go with both RNA and DNA, which ones are only RNA, or which ones are only DNA. That would be a really common thing to ask. Yes, you can. Now, last sort of thing on the docket for today is just a little bit of information about DNA sequencing, because we're going to talk a lot about DNA and the code. You already kind of know that your DNA has these things called genes. What genes are is really just a little section of DNA that directs the production of one or more polypeptides. Polypeptide is a fancy word. Poly means many, and peptide is what we call the bond between two amino acids. So what we're really saying is many amino acid bonds. It's a fancy word for protein, but I like to make sure I use both terms because I never know which term the diploma people are going to feel like using, and we're talking about the same thing. A protein is a polypeptide. A polypeptide is a protein, okay? And because of this, this is what differentiates cells. It's what makes them look different. Having a gene means you have some instructions to make a protein. When your cell makes that protein, then you get features that make one cell or one structure different from the other. Now, quite a few people have corroborated this, but Thomas Morgan is a guy who maybe first or long ago stated that genes are on chromosomes. And so people were figuring out different things in different ways, and when we put it all together, we know that we have chromosomes. We know that chromosomes are replicated and separate during cell division. We know that from studies, DNA, which chromosomes are made of, is what gets passed on. And we know that there are genes on each one of these chromosomes. So we're putting together all the pieces of how stuff gets passed on. There's another word that you might have heard before, and it's the word genome. Genome is what we use to describe the sum of all of the DNA in each cell. That includes genes and non-coding sections. You actually have a bunch of DNA in between your genes that doesn't appear to be doing anything. And I say doesn't appear because I'm sure we'll discover something later in the future, and then people in 100 years will be like, haha, look at those silly people back in 2021. Look at what they thought. But for now, it appears as though there are sections of your DNA that are not coding for anything. They're not directing your cell to produce one or more protein or polypeptide. We also know that genes are not evenly spaced on chromosomes. So it's not like there's a gene, and then there's 10 nucleotides that do nothing, and then another gene, and then 10. Some genes are really close together, and some of them are separated by like hundreds or thousands of nucleotides that appear to be doing no coding at all. And so a big area of research is DNA sequencing. There are lots of researchers that spend their whole entire career trying to figure out the DNA sequence of a particular organism. Humans, various other animals, but it's a big deal. We're trying to make a bank of what the sequence of DNA for all organisms is, and it's going to take probably forever. But it's the process of determining the exact nucleotide sequence. So in which order do the A, T, C, and G go? Maybe you've heard of the Human Genome Project.